Hey, Emma,
Hello.
how much pain are you in?
It's not great right now.
Yeah, I wondered if you might be overdoing it, but you know, A for effort.
Yeah, I'm trying to slowly get back into it.
It is what it is. You had major surgery, so give yourself time. Everyone say hi to Emma. Class says hi. All right, let's get with it here. All right, folks. In the spirit of trying to stay on schedule, you might notice up here we have podcasts, and we have many of these upcoming lectures podcasted. So we're on cancer biology. This one starts at metastasis. That's about how far I will get today. And then I will not cover the chapter on sleep and pain. And then there's the eyes as well. So I will essentially pass go, collect $200 and go straight to brain trauma after that. So if you could watch these others, these are about 10, 12, 13-minute presentations. So they have the PowerPoints with the recording. They do work, unlike some of the others that I had to redo. So today, we'll start here. I'll get up to metastasis, and then we'll jump to here, chapter 15 here. Sorry to have to do this to you, but the nature of the beast is that we just have a lot of material, and some of it I just have to cover using these sorts of techniques. So, that means you guys have to read and listen a lot to stuff, but well, you're getting your money's worth. So, all right. Hope you guys had a great weekend. Hope you enjoyed the warm weather we had. Monday morning, not Monday, Saturday morning was witheringly cold. And then it warmed up, got over 40, was actually pleasant. So I was in Clarkston. My quizzers actually remembered something. They did pretty well. My young teen vet quizzers won their division, yay team. And two of my quizzers tied for the first place for the Kahoot quizzing. And then Hezekiah Jen, he won individuals. So it's a pretty good day. Pretty good day. So all things considered, not bad. All right, we were looking at cancer. So let's do some review here. I'm just going to pick on people one at a time. So I pick you, it's not because I don't like you, it's because I just happen to random. So Maria, you define hyperplasia. You can use your notes. Go ahead. Too many cells was overgrowth. Overgrowth of cells. Overproliferation of cells. So this hyperplasia is it synonymous with cancer? No. No. Jay, why? Why is hyperplasia not the same as cancer? Good job. They just overgrow. They don't invade other tissues. When we talk about cancer, we usually mean a disease where you have overgrowth and those overgrown cells invade or impinge on other tissues. So not the same. Not the same. Miss Haley, can you define neoplasm? Neoplasm. New growth. New growth. And Liesel, that new growth has some funny things about it, right? Give me one funny thing about that new growth, that neoplasia. Abnormal growth. Do the cells look normal? They might, but they might not. So, Kirsten, benign and malignant. If I hand you a slide and tell you there's a thing on it, and you look at it, what are some things you're going to look to see if it's benign? Capsule, fibrous capsule around it. Very good. Fibrous capsule. Fibrous capsule. And Kylie, which grows faster, benign or malignant cell? Malignant, almost always. Almost always. There's some tumors that are just slow growing. They take years to grow. There's a T-cell lymphoma that tends to affect the skin. It's called mycosis fungoides. It can grow for years. Starts as a little patch, and pretty soon, after 30 years, it practically covers your whole skin. But it took 30 years to grow that fast. Very slow growth. Prostate cancer, slow growth. Other cancers, glioblastomas, for example, in the brain, not slow growing. And we took a look at some of the things that characterize these cells. We stopped last time, I believe, at this whole tumor promoting inflammation thing. Well, no, the enabling replicative immortality. And now we go here, tumor promoting inflammation. So very often tumor cells will recruit nearby cells to help them out. They will secrete certain types of either growth factors or cytokines or both, or something like that. And they'll get nearby cells to come and help them out. They will get nearby EPCs to make new blood vessels for them. They'll get fibroblasts to lay down substrate that the tumor cells can move upon. They will get other cells to surround them and wall them off from the immune response. They will recruit other cells to do their bidding, if you will. Pretty insidious. Now, this making new blood vessels, that's called angiogenesis. Angiogenesis. This is a characteristic of tumors that will spread because it gives them a highway through which to spread, but they induce those new blood vessels. The other reason why this is important for the life of the tumor is that if you have a solid tumor, full of cells like that, the cells deep in the center, they're pretty deprived of oxygen. The cells on the outside, they might have more access to oxygen and nutrients and things like that. So it's not uncommon for the big tumorous mass to have cells in the center either become necrotic or just be really, really slow growing. And the only way this tumor is really going to do something interesting from its perspective is to summon a blood vessel into it to provide blood. So these cells, these tumors, they will secrete vascular endothelial growth factor, VEGF, and they'll cause the blood system to remodel itself. So if this is a blood vessel here, you'll actually see this. It grows a little bit, then a little more, pretty soon it's inside the tumor, and then you've got another vessel going through the tumor that links up to another blood vessel, and you've got a tumor now with this wonderful blood vessel. It can grow faster, and it can invade this tumor, this blood vessel, and go to other places in the body. That's called angiogenesis. Some of the drugs that are used as adjuvants to treat tumors inhibit this angiogenesis. Any of you heard of a drug called Avastin? Avastin is a monoclonal antibody against VEGF. It's an antibody called abacazumab. You can see why they call it Avastin, because abacazumab didn't quite work. But it inhibits VEGF and prevents this angiogenesis. So they also invade other tissues. One of the ways they do that is they break down basement membranes so they can move through them. To do this, they will make enzymes called matrix metalloproteases. They're usually abbreviated MMPs. These are made early in development. They're also made during healing. You get this big fiber clot, those fibroblasts need to get through there. Oh, how do I do that? I punch holes in my clot by making matrix metalloproteases. Tumors will express these at high levels, so they degrade barriers to their invasion. They poke holes in the barriers when they scurry through those barriers. Genome instability, this is huge. If you look at chromosome smears of tumor cells, they have abnormal numbers of chromosomes. They'll also have chromosome fragments sometimes, or they may have chromosomes with big pieces of them missing. One of the reasons for this is that tumors will get rid of a gene that encodes a protein called P53. P53 is often called the guardian of the genome. P53 is involved with counting chromosomes, making sure there's a reasonable number of them. P53 gets in the way of tumor cells because they want to divide faster and faster and faster. Who cares about chromosome numbers? Not all the genes I need to do what I need, I'm just going to divide. And so they dump P53. So deletions of the P53 gene are usually indicative of a more dangerous tumor. And in fact, Bert Vogelstein at MIT has shown that in colorectal carcinoma, P53 loss is one of the steps that creates essentially a more dangerous adenoma, because it starts out as an adenoma, polyp. There's a stage 1, stage 2, stage 3, stage 4, before it becomes a full-blown colorectal carcinoma. And the transition from 2 to 3 involves the loss of P53. Resisting cell death. There's a series of genes in the genome of our cells that encode proteins that are part of this programmed cell death pathway. This is also called apoptosis. When a cell gets so irreparably damaged, this pathway gets kicked into high gear, and it's usually induced by leakage of the mitochondria. The mitochondria will leak this small protein, cytochrome C, and cytochrome C helps activate this pathway. This pathway was first elucidated in the fruit fly and also in the worm, Caenorhabditis elegans. Turns out that the genes involved with cell death in the fruit fly and the worm also are involved with cell death in our bodies. So we see how basic research can provide remarkable insights to our own cells, our own development, our own way of doing it. What you have in common with the fruit fly, genetically speaking, quite a bit. So my thesis organism was the fruit fly. My bias is fruit fly. So this pathway, this apoptotic pathway, what it does is it takes a cell, chops it up, packages it into little membrane blebs, and then throws them out of the cell. And if you see a cell that's just blebbing, that's a cell that's dying. So why does it do this? Remember what we said before? You get a bunch of cell death. Remember how that floods the body with calcium and other things, nucleotides, things like that? They're not very good for you. By bleeding and rolling off little blobs like this, the cell dies honorably without hurting anybody else. That's a silly way to put it, okay? If it works for you, we'll use that. This programmed cell death pathway in cancer cells has been hamstrung. So you can beat the cancer cell up and it won't off itself. And that's why these cells, when you put them out, when you culture, they're immortal. Just grow, 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 grow. Deregulating cellular energetics. I don't know if any of you have known anyone with cancer, or if any of you are seen as if you taking care, or if any of you home care be taking care of someone with cancer. Does anyone? How much energy do cancer patients have? Okay, Kirsten's shaking her head. Kylie's kind of going, not very much, right? The reason for that is that because the mitochondria are black in cancer cells, they're very dependent on glycolysis. The cancer cells are pigs for glucose. You do a PET scan, which the PET scan, you take 2-deoxyglucose and you put a very, very low activity radioactive isotope in it. The PET scan, essentially where everywhere in the body that's black, except for usually the bladder, that's where you have a tumor. Because this cancer cell is so stealing all the glucose from you is really, really dependent on glycolysis. And this has led people to suggest that the keto diet can actually benefit cancer patients because you're robbing the tumor of its primary food source. Many of you think that's still in the paper and magazines. Okay, I'm saying one of them as well. If you look at like Runner's World or heck, there was an article about it in Teen Vogue, for goodness sakes. That's how widespread the writing was of articles. The local paper had an article about keto and cancer. The New York Times did, the Wall Street Journal did. The evidence for this is mixed, but with certain types of chemotherapy regimens for certain types of tumors, many oncologists now are strongly suggesting the patient go on a keto diet. Because keto diets apparently will help certain types of chemotherapeutic regimens because tumor cells, they need their glucose. They can't really use other resources very well. And I don't know if that was suggested in people you take care of. No, no. Some cancer patients have experienced great results from it. Others not so much, so the data is next. No stress. Okay. Sustaining proliferative signaling, as we said, this signaling to divide. The brakes have been, the brake lines have been cut. So you just have this constant signal, grow, grow, divide, 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 divide. Within tumors, there's intense competition for cells to outgrow each other. So you get mutations occurring in cells that control cell growth. Those genes get better, better, better for promoting cell growth. And so they evade their growth suppressors, and then they will express proteins on their surface that actually inactivate immune cells that come near. So which of the following statements is true? All neoplasms are cancerous. Benign growths are cancerous. Malignant tumors have slow growth. Cancer refers to a malignant tumor. Which of those statements do you think is true? Is A true? No, okay, it's about 4 or 5 of you shake your heads. The others of you to cautious to do such a thing. Benign growths are cancerous. That true? No, more of you are shaking your head, so that really must not be true. Malignant tumors have slow growth. Emma, do you think that's true?
No.
No? Emma says no. Anyone disagree with Emma? No? D, cancer refers to a malignant tumor. All right, yes, that's the one that's true. Cancer refers to a malignant tumor. Benign tumors are not invasive, they're not cancerous. Malignant tumors have rapid growth and neoplasms can be benign or cancerous. So because you have to have multiple mutations to create a cancer cell, that takes time. Cancer, therefore, predominantly is a disease of the aged. When you look at a tumor, it's clear that it is descended from a single cell. So those cells are clonal. Having said that, the cells descended from that single cell will acquire additional mutations that give them growth advantages. So when a cell goes from a normal cell to a tumor cell, that process is called transformation. Transformation. This has been heavily studied in culture. The original people were the Weinstein lab at, where was Weinstein? Not Amherst, Harvard, he was at Harvard. So they have decreased need for growth factors to multiply. The signal transduction pathways put in motion by those growth factors are already perpetually on. They lack contact inhibition. If you take tongue cells from a mouse, put them in culture, those cells will spread out, grow. They'll fill the dish. When they're in contact with each other, they'll stop growing. Cancer cells will pile up on top of each other. This contact inhibition is inhibited. Anchorage dependence that you see in tissue culture cells is abrogated in cancer cells. That's why there's an assay that's done called soft auger assay. Normal cells will not grow in soft auger. There's no place for them to attach. Cancer cells grow beautifully in soft auger because they have anchorage independence. Or immortality. Normal cells, they get to that Hayflick limit, they stop growing. Not cancer cells, they just burn right through it, keep growing. So we talked about this before. Just mention it very quickly. Proto-oncogenes. These are genes that direct growth. Direct growth. These genes typically are regulated in some way. Oncogenes are mutant versions of proto-oncogenes. That regulation has been dismissed. Cancer cells will have one or more, usually more, oncogenes. Tumor suppressor genes. They encode proteins that down-regulate cell proliferation. Tumor cells will have loss of function mutations in tumor suppressor genes. So how does one turn a proto-oncogene into an oncogene? Turns out there's several ways to skin that cat, shall we say, to make reference to your NPS. Point mutations can do it in some cases. So the RAS, RAS oncogene. This is a small GTP binding protein. Binds GTP, gets activated, does its thing, and then after doing its thing for a little bit, it takes that GTP and clips it to a GDP plus phosphate, inorganic phosphate, and it's not activated. These point mutations in RAS make it so that it doesn't hydrolyze GTP anymore. It binds GTP, And just keep going, it becomes the ever ready button. Keep going and going and going and going and going and going. Other ways to convert proto-oncogenes into oncogenes is gene amplification, that is, you make lots of copies of it. One of the ways you do this is with the MYC proto-oncogene. In human neuroblastomas, there are multiple copies of the MYC gene, thereby making enormous amounts of the MYC protein. Those enormous amounts of the MYC protein drive the growth of neurons, creating tumors in the brain. Other ways you can do this is through chromosomal translocations, where you take a piece of one chromosome, you break it, and then you swap it with another chromosome that's had another double-stranded break in it. This cutting and pasting, if you will, of non-homologous chromosomes can generate genes that encode products that have lost their controls on them. So Burkitt's lymphoma, as we will see, involves a translocation. So does chronic myelogenous leukemia. That involves the production of a new gene product that has two proto-oncogenes that are now linked together. And by linking them together, you make this fusion protein that has no regulation over its activity. So translocations are another way to do this. So translocations can cause the production of a chimeric protein that's hybrid. It's a little bit country, a little bit rock and roll. You put it together. It's a little bit crunk and crawl, shall we say. And that protein now has no regulation, or it just gets overexpressed because of a new promoter. Or gene amplification or through point mutations. Those are ways essentially you can do that. You can also convert proto-oncogenes into oncogenes by removing pieces that are necessary for the protein to regulate itself. That's another way you can do that. Loss of function mutations in tumor suppressor genes like p53, like the retinoblastoma gene. These can do away with controls on cell proliferation. And then genomic instability is due to deletion or loss of function mutations in so-called caretaker genes, of which p53 is one of them, that encode for proteins involved either in repairing damaged DNA or genes that encode products that arrest the cell in the cell cycle if the genome integrity is compromised. So because of this, you get chromosomal instability, you get chromosome loss, chromosome amplification, loss of heterozygosity. And to show you an example of this, here are some stains. So on your left, you have a cell that is stained with a specific probe for a specific chromosome. Notice there are two copies of that chromosome, just as it should be. Use the same probe on a tumor cell. Now tell me what you see. There are a whole lot of copies of that particular gene, aren't there? on lots of different chromosomes or chromosome fragments. So that's a cell whose genome is very, very unstable. And this has been shown in lots and lots and lots of different cell lines. So cells from our bodies are not immortal. They only divide a limited number of times. The tips of our chromosomes are called telomeres. There's a special enzyme for replicating the tips of our chromosomes. That enzyme is called telomerase. Telomerase at your age is pretty robust, but as you age, its activity is going to start to diminish. At my age, it's pretty low, so I don't heal as fast as you do. Tumor cells, however, activate their telomerase. That's why tumor cells can divide over and over and over and over and over again. So this business about chromosomes shrinking, which happens as you get older, that's not the case in tumor cells because they upregulate this telomerase. And telomerase, it turns out, is a target for anti-cancer drugs. And the drug imetelstat, for example, is an anti-telomerase medication. Imetelstat is usually given to people who are elderly, who don't have much telomerase activity in the first place. So imetelstat tends to inhibit cancer cells only, not normal cells. And this simply shows you the telomeres, those caps at the end of chromosomes, the telomerase that is used to replicate them. Telomerase is low except in certain stem cell populations, like in your bone marrow, like in your tooth pits, like in other places of your body. Angiogenesis is the result of the secretion of angiogenic factors like VEGF, vascular endothelial growth factor, platelet-derived growth factor, basic fibroblast growth factor, GF means growth factor. All of those can facilitate the remodeling of the vascular system to bring new blood vessels to the tumor. And this is a picture stolen shamelessly from your book. And you can see essentially because the tumor, there it is. The tumor here is secreting these growth factors. These endothelial cells in the blood vessels have receptors for these growth factors. And in response to activation by those growth factors, they will start to proliferate and integrate with other cells that come in from the bone marrow, EPCs and things like that, to be able to come to the tumor and actually bring a blood supply to them. So decades ago, there was a German scientist whose name was Warburg. And Warburg was interested in finding a way to get tumor cells to survive outside the body. He was one of the pioneers of tissue culture. And when he did this, he discovered that the more sugar he threw in with the cells, the faster they would grow. And this was about the time it was clear there was this disease that caused certain people's urine to smell fruity. Anybody know what that disease might be? Diabetes. So he took the blood of these people who had this fruity smelling urine, didn't know what diabetes was, and he filtered it and he put it in with these cells, and these cells grew as well as if he gave them glucose. He didn't make the connection because he didn't know about insulin. Banting and Best were the ones who discovered insulin about 40 years after this chap. So this was called the Warburg effect. Namely, the tumor cells really needed a lot of glucose. And the products of glycolysis provide ATP via substrate level phosphorylation. Turns out many of those genes that encode glycolytic enzymes are activated by oncogenes. And this is the very reason why PET scans work to detect tumors, because of their very high glucose metabolism. I always get asked this question, so I will try and preempt it. Will a keto diet keep you from getting cancer? There's no evidence of that. And it has been researched. So there's no evidence for that. If you feel like eating keto makes you feel better, please eat all the keto you want. But please don't claim it prevents you from getting cancer, because there's no evidence for that. Maybe in the future there will be, but there's none right now. Are any of you in nutrition right now? Has Beth said anything about the keto diet and cancer? Okay, there is quite a bit of work actually ongoing right now. If you go to clinicaltrials.gov and you type in keto diet, it's a long list. If you do keto diet cancer, that list only gets a little bit shorter. So there's a lot of keto diet research going on now. And no one has shown that it prevents you from getting cancer. So, all right. So as you can see, in normal cells, glucose is brought in response to insulin by facilitated diffusion. So this is the GLUT4 facilitated diffusion transporter that you find largely in liver, skeletal muscle, and adipose tissue. Excuse me. Other cells will use this GLUT2 facilitated diffusion transporter. They can take up glucose, but not as quickly. And they will convert glucose via glycolysis to pyruvate. Pyruvate will go to the mitochondrion. It'll be converted to acetyl coenzyme A. Acetyl coenzyme A enters the Krebs cycle, gets completely oxidized to carbon dioxide and water. The electrons are then used for oxidative phosphorylation to make ATP, blah, 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 blah. You know the story. In exercising muscle, particularly fast twitch muscle, you don't have as many mitochondria, so the reductant that you generate, NADH, that you generate via glycolysis, you have to regenerate it. To do that, you dump the NADH here on the pyruvate. When you reduce pyruvate, you make lactate. Don't believe me? That's pyruvate. You reduce it, you get lactate. So those electrons that you generate during glycolysis and what's the enzyme that catalyzes this reaction? Anybody remember? Lactate dehydrogenase. That's an important marker for certain types of pathologic conditions, like muscle damage. It's an important liver marker. That's an enzyme you should remember. Lactate dehydrogenase. In cancer cells, the mitochondria is whack. Because the mitochondria is whack, you're going to depend a lot more on glycolysis. So you're going to produce a good deal of lactate. Now, lactate can be recycled by which organ? Liver, via gluconeogenesis to make sugar. So lactate can be recycled, But if you have a large tumor load, you're going to have metabolic acidosis. And people with advanced cancer, that's a second reason why they're so tired all the time. They can eat all they want, but it's being chewed up by the tumor, and the tumor is giving them lots of lactate. And then the cells resist apoptosis. So this apoptotic pathway is very complicated. Here's the take-home message from this. This can be induced either intrinsically with damage, so DNA damage or mitochondrial damage will do it, or it can be induced extrinsically. Extrinsically, there are receptors on the surfaces of cells that tell those cells to off themselves. And members of this particular family, the FAS family, bind those receptors. A member of the FAS family is tumor necrosis factor. And then black box, antimiracle occurs and you get apoptosis. We could go into the mediators here, but I don't want to. That's beyond the scope of this class. Cancer cells have hamstrung. So these particular factors here that resist apoptosis, they will upregulate these, for example, BCL2. BCL2 is very commonly upregulated in leukemias, for example. They will upregulate, for example, this protein here, IAP, that prevents initiation, essentially, of this process. They'll do all manner of things to prevent this from happening. Now, chronic inflammation has been pretty inextricably linked to cancer. For example, what if you have chronic inflammation in the GI tract? Say you have an IBD, an inflammatory bowel disease, like Crohn's disease, like ulcerative colitis. You're at higher risk for colorectal carcinoma. What if you have chronic inflammation of the liver, as in the case of chronic hepatitis C, or chronic hepatitis B, or cirrhosis, or NASH, or something like that? You are at increased risk for hepatocellular carcinoma. What if you have chronic inflammation in the kidney because of Buerger's disease or ingestion of certain types of toxins or something like that? You're at chronic risk for RCC, renal cell carcinoma, and so on and so forth. The reason for this is that there's constant cell damage and constant cell proliferation to regenerate those cells. In the constant cell proliferation, you have these cells that keep dividing. You have more opportunities for mutations that will transform those cells into cancer cells. So chronic inflammation is inextricably linked to higher risk of cancer. And I hope that makes some intuitive sense to you. Given that increased cell division, like with aging, gives you increased opportunities for mutations that can cause cancer. Krishna, you don't look convinced. You're sitting there going, I'm not sure I believe that. You believe it? You look very skeptical. Skepticism is not a bad thing. Not a bad thing, but I'm just wondering what does it take to convince you? You're just being pensive. You're just tired. Okay, all right. Well, we have that in common. Now, for example, Helicobacter pylori we'll discover next year in micro. It's a microorganism. It causes a chronic inflammation, chronic infection of the GI, the stomach mucosa. Because you get this constant damage, replacement, damage, replacement, damage, replacement, you can get ulcers. And then you divide to repair that ulcer. This can predispose you to carcinoma of the stomach or mucosal associated lymphoid tissue lymphomas, because those gastric associated lymphoid tissue they're constantly having to divide to make cells that can fight and secrete antibodies that can fight the Helicobacter pylori. So Helicobacter pylori disease can predispose you to either one of those two tumors, stomach carcinoma or the MALT lymphomas. There are also other cells that are recruited by the tumor, and one of them is macrophages. They recruit the macrophages, and these macrophages have a kind of pro-healing phenotype. And the more of these you have, it turns out the worse your prognosis is. These cells have a diminished ability to phagocytose and deal with invaders. They have diminished cytotoxic response, but they also secrete cytokines that suppress the immune system. They suppress the immune response to the tumor. And it's because of these TAMs, tumor-associated macrophages. Now, typically, the immune system undergoes surveillance, and this surveillance will prevent tumors from arising. Experiments in the mouse, for example, have shown that there are certain tumors of the aged in the mouse. You can actually take antigens, surface antigens from those tumors. You can inoculate mice with them, and essentially you can vaccinate mice against these tumors of the aged, because the immune system undergoes this constant immune surveillance. And it's also clear that as you get older, that immune surveillance starts to diminish. And that might be one of the reasons why you have more tumors that arise when you get older. Now, some people, because they have autoimmune diseases, will take drugs that suppress the immune system. You'll learn about those next year in Pharm. There are monoclonal antibodies that are used for against tumor necrosis factor. Drugs like infliximab, drugs like adalimumab, drugs like certolizumab, so on and so forth. There are other drugs that will bind and inhibit interleukin-6. Some of these were actually used to treat COVID-19. Little bit of tocilizumab, for example, sarilumab, drugs like that. There are also drugs that can inhibit B cells, that they bind to a surface protein called CD20. You emailed me about one of them. What's that called? Rituximab. Rituximab. Grace is an expert on rituximab. She's been doing some research on it. And how does it work? Right. Binds to the surface of B cells, and it causes the immune system to recognize those B cells, destroy them. So you don't make these antibodies that might be messing with your system or something like that. When you get on these drugs, you are told by the rheumatologist, this is going to increase your risk for cancer. And the clinical data, the post-marketing data on those drugs clearly shows you suppress the immune response, you increase the risk of cancer. Same thing with steroids, they increase the risk for cancer. Biologics, other types of drugs, methotrexate, azathioprine, other types of medications that do the same sorts of things. They will increase your risk of things like non-Hodgkin lymphoma by about 10-fold. They can increase the risk of a skin cancer called Kaposi sarcoma by 1,000-fold. And tumors, in fact, will release immunosuppressive molecules into your blood, and that will decrease the immune system's ability to recognize them and make them resistant to chemotherapy and radiotherapy as well. Because one of the things chemotherapy and radiotherapy does is it damages the tumor cells. So now these other cells, especially natural killer cells, come in and say, okay, you're packing it up, pal. And the tumor cell essentially kind of holds up a sign that says, no, you can't. And then the cell backs off. So again, this is stolen from your book, but you can see that the tumor cell will express tumor antigens that are recognized by T cytotoxic cells and natural killer cells, but they may not produce that tumor antigen. Now the T cell can't recognize them. Or they might not make class 1 proteins because they don't make class 1 MHCs. T cells can't recognize them. Or they might make inhibitory ligands like PD-1 that actually inhibit those T cells from actually recognizing and acting against them. Now, it was first shown in animals, but it's very clear from work in humans as well. Certain viruses can cause cancer. The way these viruses cause cancer varies from virus to virus, but hepatitis B and C viruses clearly increase your risk of hepatocellular carcinoma. Epstein-Barr virus can increase your risk of Burkitt's lymphoma, particularly in people who are of African descent. Kaposi sarcoma herpesvirus. The Kaposi sarcoma herpes virus increases your risk of Kaposi sarcoma. Human papillomavirus, certain strains of it. Most of the strains just cause warts on your hands and nose and elbows or something like that. Others will cause cervical carcinomas of the cervix. And then HTLV1 and 2 will cause human T cell lymphomas. The way they do this is either some of these viruses actually import their own oncogenes into the cell. Others promote a constant low-level inflammation. That seems to be how hepatitis C works. And by promoting this constant low-level inflammation, get regeneration, constant division, constant introduction of immune mutations, so on and so forth. HTLV1 and 2 have these sequences. They will plop down next to a proto-oncogene and alter its regulation. They now upregulate that gene product. And that's how you're able to get more of that proto-oncogene promoting cell division. Human papillomavirus has a protein in its genome that it encodes called E7. E7 inactivates tumor suppressor genes. So these viruses have lots of tricks up their sleeves. But infection with these viruses can increase your risk of certain tumors. And it is here that the recording takes over. So I am going to let you listen to the metastasis recording. And in the few minutes we have left, oh, mercy, what did I do wrong? Okay, did I not? Nope, I didn't. Yes, discard. Okay. So now I need to go back here and then go here. All right. So the brain is the organ that's responsible for learning and memory. It's responsible for most of the aspects of your personality, and it's also responsible for consciousness. Now, we could have a very spirited discussion about your brain and your soul, whether they're the same thing or something like that. Kind of beyond the scope of this class, I think it's pretty clear that your spirit, your soul, uses your brain instrumentally to get certain things done. And without a functioning brain, your spirit or soul can't really use your body to do what it wants to do. Because we just quizzed over it, I'm very cognizant of the story of the Gadarene demoniacs. And Jesus gets to the other side of the Sea of Galilee and the Decapolis on the eastern side of the Sea of Galilee, and the text says that 2 demon-possessed men came up from the tombs to meet him. They were so violent that no one could pass that way. And they shouted at him, "What have you to do with us, Son of God? Have you come to torture us before the appointed time?" Now, these are obviously 2 chaps who are not in their right mind. First of all, who in their right mind chooses to live in a tomb? You? No, did you? Did you? Did you? And they're shouting and screaming all kinds of weird things, and what have you. So, yeah, not in their right minds. Now, were their brains diseased? Perhaps, but something else was controlling their brains. Because we know in other gospels that when the people of the town went out to see them, he was clothed and in his right mind. And he wanted to go with Jesus when the town people of Decapolis told him, Jesus, just go away, please. You deprived us of our pigs and we're mad at you. So the brain is used by your soul, your spirit, your whatever, and without it, you're not conscious. Now, there's a way to clinically, objectively determine consciousness. You have a handout that is in back. I hope you picked it up. Called the Glasgow Coma Scale. You guys will have a lot of exposure to this. Anything that do either emergency or ICU nursing will be very, very well acquainted with the Glasgow Coma Scale. You'll be able to do this in your sleep. And you'll notice that the Glasgow Coma Scale uses 3 different categories, essentially, to ascertain consciousness. Marie, what's the first one? You see there.
Eye-opening response.
Okay, eye-opening response. The eye-opening response is in response to just you coming into the room. Now, if they don't open their eyes when you come into the room, you'll learn in the other courses you're supposed to come in, wash your hands, and then introduce yourself. What's the first things you do? Not necessarily in that order. You may want to introduce yourself and then wash your hands, but or you may want to wash your hands outside and you go, oh, can't do that because you can't do it. Oh, better wash your hands inside. Because you have to have clean hands for doing the rotation. But if they open their eyes, because usually when you come in, pull the curtain or come in, close the door, something like that, you're making noise. Usually they open their eyes, if they have to go slow. And if they don't open their eyes instantly, then what are you going to do? Wake up! What are you going to do? You have to open their eyes. Yeah, not wake up, don't do that. You get fired. One trick you can do is you go above the eyebrow and you just stroke that like that. Just stroke the eyelid. And that's gentle, but if they open their eye in response to that, you lean over, you have their charts, you know their name, you say their name, you say it gently, and then you will stroke right here. When you do that, you can say, go do it yourself. You can see it's not unpleasant, it doesn't hurt or anything. But if someone did that to you, you probably won't fight. Something like that. So the stroking, that's another way to get them to open their eyes. If that doesn't work, what are you supposed to do that? Yeah. Okay, painful stimuli. You're just going to take the finger and you're just going to get a little, just squeeze just a little. You're not going to take a pair of pliers and sit there and just take the finger and we'll squeeze. Or you can even pinch. Just like that. You don't have to hurt the patient. You just get a little painful stimuli. Well, and then they may either draw it back or not open their eyes at all. They open their eyes. That's usually point. You just some sort of painful stimulus, doesn't have to be, okay, Jake, what's next one? Well, after the eye opening, what's next one? Verbal response. Verbal response, you're gonna ask the patient questions. What kind of questions you gonna ask them? Yeah, your name, date, you know who you are, you know what happened. If it's an accident, head trauma, they won't remember the accident, but they might remember what happened just before. Oh, I was driving and I got to such and such an intersection. And then I just remember the car spinning or something like that, or what have you. Or they might not remember anything. Or they might not be able to form words. What other responses might be there?
Inappropriate word.
Inappropriate words, aphasia. Aphasia. If you can't match the right word with the right thing. They might be able to talk very fluently, but they don't call a cup a cup. They don't say, I feel fine. They'll say, I feel morning. I feel regarded. I feel just. And the brain might be a little swollen. That might be temporary. It might go away. But you need to note that essentially on the chart. And that's something that the Glasgow Coma score you're giving them is a score you're giving them right now. And you form a relationship with this patient, you may want to go easy on them. Don't, because you need to give an objective assertion of what their Glasgow score is, whether you like them or not. And that's why this is a family member, something like that, you can step out. Because your objectivity is helping, right? Reagan, right? You know, got 2 minutes about that. No, your objectivity is one of the best things you can give to a patient. You need to be objective when you walk into that room. All right, we will continue with this. Next time, we'll get into alterations in cognitive systems, movement disorders, dementia, and the like. Yeah, that's all recorded. 12, 13 minutes. Okay, Emma, don't overdo yourself. Don't overdo it, okay? Heal thyself, all right?
Yeah, I'm trying. I just have a quick question before
Yeah.
I head out. So I'm looking at I was looking at the grades last night, and I emailed you in quiz number 2, and it's not in
No,
Blackboard.
I had a
just
busy
didn't know if-
weekend, so I wasn't able to get in and record stuff during the weekend.
Okay, perfect. I just didn't know if you didn't get it or
No,
if
I got
ended
it.
up-
I just haven't
Okay.
been able to do anything with it yet.
Okay, no problem. Sorry. I was just making sure.
Okay,
All right, have a good day.
you too, Emma.
Bye.